we just published a study in Science looking at overcoming BTK-resistant mutations in chronic lymphocytic leukemia by using BTK-degrader therapies. So the overall design of this study was that it was a combination of laboratory research as well as part of a phase 1 clinical trial. So on the laboratory research, we had recently defined mutations in BTK, Bruton's tyrosine kinase, that is the target of BTK inhibitors that are used most commonly for CLL, but also other types of lymphoma. We had discovered these mutations that cause resistance to a class of BTK inhibitors called noncovalent inhibitors. And in understanding how these mutations work, we discovered that some of them cause a new function of BTK. They actually decrease the kinase activity, but increase the ability of BTK to act as a scaffold for other kinases to come and bind to and increase signaling in the leukemia cells, thus making patients resistant to the noncovalent BTK inhibitors and other BTK inhibitors. So in the second part of the study, we partnered with Nurix Therapeutics to use a BTK degrader called NX-2127, which is currently being used in clinical trials. And we used it in our laboratory setting to show that by degrading BTK, which is essentially using this molecule where one part of it attaches to BTK and the other part attaches to the cell's machinery, the proteasome that degrades proteins, you're able to degrade selectively BTK as well as IKZF1 and 3 with this particular molecule. But we showed in the cell lines that despite which mutation, whether it was one of our new mutations or mutations that cause resistance to covalent BTK inhibitors like ibrutinib and acalabrutinib, we were able to degrade BTK regardless of the mutation type and kill the CLL cells. And excitingly, because Nurix was testing this in a phase 1 study in CLL patients as well as other patients with non-Hodgkin's lymphoma, we had samples from those patients. Some of them, all of them had been on previous BTK inhibitors, so some of them had mutations in BTK, and we were able to see in the patient samples themselves, patients taking NX-2127 had BTK degradation independent of the mutation status, as well as responses independent of the mutation status at this particular stage of the phase 1 clinical trial. What's next for us in this line of research is we did work to define the specific kinases that are binding to BTK now when it acts as a scaffold. We'd like to further understand exactly how those are binding to BTK, and potentially there could be a way to block that binding other than just degrading BTK, or those particular kinases that we defined, such as hematopoietic cell kinase HCK and integrin link kinase ILK. Perhaps those could be directly targeted either with BTK or alone in these mutant cells as another type of therapy to overcome the BTK inhibitor resistance in CLL patients.